for example, what 3 different products were made in response to very specified RNA sequence. Right? And it wasn't just actually a triplet repeat. The experiments were done by Golpanian, and he also used repeat of 4 nucleotides. He got some unusual results there. Good. All right. So we know those. We understand the 2 chemical reactions that are catalyzed by the amino-acyl tRNA synthetases, and I will review this for you in just a moment. We also understand why the activities of these amino-acyl tRNA synthetases are sometimes described as a second genetic code, and we can understand that the amino-acyl tRNA synthetases are really the guardian of fidelity in the translation. They need to get it right, and so we understand that they have kind of a mechanism which first screens for the proper amino acid to conjugate it onto the tRNA, and then does an editing to remove an improperly amino-acylated species if it doesn't belong in that particular tRNA. Okay, so those are the big topics on Tuesday. Great. So in the spirit of making sure that we know the critical topics from last time, here are these 2 sequential reactions that are being catalyzed by the amino-acyl tRNA synthetases. They're looking for a particular amino acid, 1 of the 20, and so this explains why this is a family of synthetases. There's essentially 20 of these enzymes, 1 for each of the natural amino acids. They're going to use this as their substrate, and the other substrate for all of these reactions is an ATP. And the first step is to activate that amino acid by turning it into an amino-acyl adenylase. Right? So you should be able to draw out this chemical reaction. Note that the carboxylate group is being used as the nucleophile. This target of the nucleophilic attack is one of our favorite targets, the alpha phosphate of a nucleotide triphosphate, and that's going to create this phosphoanhydride, this amino-acyl adenylate. Now that we have this activated species, we can conjugate it onto the 3' position of the terminal 3' adenine on a tRNA. Right? So each one of the different tRNAs gets its own particular amino acid at that 3' and on the CCA arm, and the amino-acyl tRNA synthetase needs to get that right. Okay. Great. So the amino-acyl tRNA synthetases really need to do 2 jobs. They need to recognize the proper tRNA, and there's like anywhere 30 to 40 different tRNAs. Needs to make sure it grabs one of the proper ones. It also needs to make sure that it grabs the proper amino acid and activates it and attaches it onto the tRNA. We talked before about the fact that in order to discriminate the proper tRNAs, the enzyme is looking at the tRNAs, and it's usually using structural information, the fold of the tRNA. It's looking in the anticodon loop to make sure it's the right one. These have got to be distinct in that anticodon loop. And then it's using all that information as it's hugging the tRNA to make sure it's got the right one. We also talked about these 2 different active sites on the amino-acyl tRNA synthetase, the site for activation and the site for editing off improperly conjugated species. So here is the extracrystal structure of an amino-acyl tRNA synthetase and complex with its tRNA. Here is the ATP. This defines the activation site right here. And we talked before that there's 2 different classes. These enzymes can either be monomers, dimers, in some cases tetramers, so a whole variety of different things. And both the sequence and the structure of tRNAs are being used by these synthetases to make sure that they grab the right one. All right. So let's begin thinking about how the ribosome is going to carry out its job. And very high level first preliminary thought is, when we took a look at synthesis of DNA polymers and RNA polymers, we talked about the fact that it was directional, 5' to 3'. And not surprisingly, synthesis of proteins is conducted in a very specific direction as well. That is, from the amino terminus to the carboxy terminus. So if you take a look at a polypeptide sequence, a protein sequence, the last one, the last residue that's placed onto that polypeptide chain by the ribosome before it produces its product is that amino acid right on the C terminus. And you won't be too surprised when I tell you that I really care about how we know that. It's not enough for you just to know amino terminus to carboxy terminus. I want you guys to know, how did they figure it out? So this was a kind of a cool experiment. It's a little confusing, so we'll take a little bit of time and we'll go over it, make sure we understand it. This experiment was carried out by a guy named Dinsis, and he was looking at the synthesis of globin chains. Pretty sure it was hemoglobin. And he was adding radioactively labeled leucines into this protein synthesis machinery. He was doing like a time course, right? If you just sprinkled in a little bit for just a few minutes and then collected the product, what would he get? Or if he just allowed this radioisotopically labeled leucine to like incubate in this protein expression system for an hour, what kind of product would he get? And in particular, what he was looking at is when he chopped up the full-length product, where was the radioactivity? Was it scattered throughout the entire protein sequence? Was it distributed more on one end than the other? And how did this vary through the different incubation times? And here's what he saw. The red here indicates where the radioisotopically labeled leucine was positioned. And if you just allowed a very short time course and then collected all the full-length proteins, he barely saw any radioactivity towards the N terminus, and it was all popping up towards the C terminus. Now, if you allowed that radio label to be conducted over a really long period of time, essentially bathing the entire protein chain, sure enough, the radio label was positioned throughout the entire chain. So what did that tell him? It told him that if you just collect the protein when it's only had enough time to make that segment of the chain at the very end of the synthesis, that the end of the synthesis was at the C terminal end. This is the part that was made last. If he incubated it for about enough time as would be required to synthesize half the chain, what half the chain was labeled? The half at the C terminus. Conclusion, this was made first, this was made last. All right, I'm going to give you a couple more slides just because sometimes when students have gone back and looked at this, get a little bit perplexed. I apologize that this is a little bit fuzzy, but this just provides another description of how this experiment was conducted. The blue here indicates unlabeled protein segments. The red indicates portions of the protein chain that have the radio label. All right, so what happens when he just collects material right at the 0 time point? Well, polypeptides are being synthesized on the ribosome, but they're different lengths, and none of them have any radio label because none of it was added for any period of time. If he added the radio label for 5 minutes, collects the protein chains, some of them partial length, some of them full length, and he only looks at the full length proteins, chops them up, analyzes the fragments that are produced, identifies which fragments are which, sees the radioisotope label only at those peptide fragments that are closer to the C-terminus. And if you allow the time course to go longer, and again, fragmented the full-length protein products, again, weighted more with the radioisotopic label at the C-terminus, and seeing a little bit of cold peptide fragments towards the N-terminus. Same conclusion. What is made last? The portion of the protein chain at the C-terminus. One more slide. Again, here shows the relative amount of tritium. This was the radio label that was being used. Different peptide fragments on the beta chain of this globin. These numbers don't necessarily mean too much in this case. Just know that the peptide fragments over here are towards the N-terminus. The peptide fragments over here are towards the C-terminus. And again, the ones that are the protein chains that are collected very quickly with a brief pulse of radiolabel, all the radioisotope is preferentially located towards the C-terminus. You still see that waiting after 7 minutes, but if you wait for an hour, essentially the entire protein chain is bathed in the radiolabel. Okay, so this was the experiment that was used to convince everybody that the ribosome is making the polypeptide chain, starting at the N-terminus, completing it at the C-terminus. Does that make sense? Okay. All right, so once again, as we think about what happens in this beautiful piece of machinery, we're going to think about these 5 different steps. We've already covered activation of the amino acids and conjugation of the amino acids onto the tRNAs. Now we're going to think about today initiation, elongation, termination, and release of the polypeptide synthesized on the ribosome. And the last step that's shown here is folding and post-translational processing. I'll just mention briefly about folding because we already did talk pretty much about it. And then I'll just touch on post-translational modifications of proteins today, and we'll get into more detail on that next week. For each of these steps, I think it's really helpful to know what are the critical components. What does the ribosome need to assemble in order to move forward with each of these steps? And so we'll be taking a look at that as we go through each one of these today. Yeah.
Sorry, do you mind going back to the previous slide for a second? So are we saying that the synthesis is happening from the C-terminus to the N-terminus?
No. The synthesis begins at the N-terminus and completes at the C-terminus.
So why would we see the radioleucine at the C-terminus?
Right. It's because if you collect only those protein chains that have been completely synthesized,
okay,
right, and if you have a brief pulse and then collect the fully synthesized protein chains, you will only have had time to sprinkle in the radiolabel on the portion of the protein chain that was made right at the end, just as the ribosome was completing the last few steps. Okay? So that tells you what was the last bit of the chain to get completed. If the radioisotope label ends up at that part of the chain, that's the last bit. Okay? So it's exactly this kind of wrapping your head around it, which is why I showed you 3 different data slides about how that experiment was conducted.
Right, thanks.
Got it. You got it. Yes?
How do we know that
It depends upon the system, like what temperature, what kind of reagents you use, how much ribosome you have, whether or not you have sufficient mRNA. It's very condition dependent. It also depends upon what kind of polypeptide chain you're making. For example, if you were making collagen, for example, that has only really 3 amino acids in it, and you don't add a sufficient amount of those tRNAs, the ribosome is going to be waiting around for those tRNAs to get recycled. So So it's very system dependent. You can, provide some kind of rough calculation about the speed at which the ribosome does its work. Like if you figured out, okay, this is a 500 amino acid protein, this is kind of telling you that the protein chain is completed after about 10 minutes. You can make that calculation and figure out how many amino acids are being added per second onto the growing polypeptide chain. But whether or not these in vitro experiments actually correspond to what's happening in the cell is kind of a different question. Yeah.
If the freeing up of tRNAs is the rate limiting component of this process, does it have a Km value associated with it, or does that not apply to this kind of situation?
Are you asking about the ribosome having a Km for the tRNAs, or the amino acyl tRNA synthetases having a Km for the tRNAs that they're charging?
Well, I would think that it's whatever is slower, or it's whatever limits the process.
Yeah.
No.
So that really will depend on what the concentrations of the tRNAs are. I'm comfortable with considering a Km for an amino acyl tRNA synthetase with regard to tRNA as a substrate. I'm having trouble thinking about how you would calculate a Km for the ribosome. The ribosome for sure is an enzymatic system. I'm not sure you could really calculate a Km. That's a good question. I really don't know. Yeah. These are like sort of arbitrary peptide identifiers. Yeah. And they just figured out later which one of these peptide numbers was closer to the N terminus or closer to the C terminus. It would have been better if this was like alpha, beta, gamma. Yeah. All right. So let's get into it. Initiation, elongation, termination. So what are the ribosomes? What are they made of? They're ribonuclear protein complexes, right? So these big pieces of machinery include both protein and ribosomal RNA. What do they chew on? They chew on mRNA as a template. They use tRNAs that are charged with amino acids as substrates, and they take these materials and they form proteins. For an E. coli cell, a huge part of the mass that's inside the cell actually is the protein synthetic machinery. This is a big part of what simple cells are doing. They're making protein. 20,000 ribosomes estimated. Each one of them is 2,700 kilodaltons, so we're talking almost 3 million molecular weight, and it's about a quarter of the mass of the cell. Very early on, even crude electron microscopy was able to show that the ribosome is composed of two subunits. We call this the 30S and the 50S, and this S stands for Svedberg unit, which is a measure of to what position a macromolecule will migrate in the presence of a centrifugal force. So this is about separating these subunits in a centrifuge. Each one of the two subunits has ribosomal RNA in it. In the smaller subunit, it's an RNA called the 16S RNA. The larger subunit has two pieces of RNA in it. There's the 23S and the 5S ribosomal RNA. And in addition to those three RNA molecules, there are specific proteins that are also used to form the E. coli ribosome, 21 in the small subunit, 34 in the large subunit. So you can see that this is a very complex piece of machinery that includes both RNA and protein. And what's amazing for me as a chemist is that you can completely take this thing apart and put it all back together. Pretty incredible. All right, so here's the whole thing. We disassociate the two subunits. Here's the small subunit, large subunit. And then we take apart the individual macromolecules from these subunits. We disassociate these things into the constituent proteins and the constituent RNA. Same thing over here, providing us two different RNAs. Now that we have all those individual components, we begin putting them back together. Reconstitute the small subunit, reconstitute the large subunit, allow the large and small subunits to come together, boom, working ribosome. Pretty amazing. Almost 3 million molecular weight, take it all apart, put it back together. All right, so let's get started with making a polypeptide on the ribosome. We are going to start where? What's that in the polypeptide chain? N-terminus. All right. So here's our N-terminus. That's where we're going to begin. And for bacterial protein synthesis, we start with this amino acid each time. When we were talking about the genetic code, we talked about the fact that the start codon was AUG. That encodes for methionine. A particular AUG codon can be recognized as the start codon. And when it's recognized as a start codon, it doesn't just use methionine, it uses this special methionine that's got a formyl group on it, N-formyl methionine. This is what is the first amino acid used for bacterial protein synthesis. So this does not look exactly what we might expect, but what the cell does is it generates a regular methionine charged onto tRNA, and for the start tRNA, it will convert that into the N-formyl methionine just by tacking on that formyl group. And then many of you, I'm sure, have expressed proteins in a bacterial host, and you probably have not, in most cases, needed to worry about this N-formyl methionine. Sometimes you don't even really care what exactly is on the N-terminus of the protein, but oftentimes you never even see it because as the protein is processed, it just lops that first amino acid off. Okay? Great. So let's think about how the ribosome knows where to get started, how to get started, what does it need to get started. It requires 3 initiation factors. These are proteins. Oh, no problem. All right, 3 initiation factors. We need GTP, this is an energy source, and we need some magnesium divalent cation. The ribosome has 3 binding sites on it for tRNAs. There is an A site, that stands for an aminoacyl site, a P site, which stands for a peptidyl site, and an E site, which isn't really depicted here because we don't properly form the E site until we bring in the large subunit. We just see it develop right down here at the E site. Okay? So the peptidyl site, that's understandable because we make our peptide bonds at that peptidyl site. That's where the bond forming events happen. That's where the catalytic center is. The aminoacyl site, the A site, okay, that's where the incoming aminoacyl tRNAs initially dock onto the ribosome. And the E site, the exit site, that makes sense. That's where the tRNAs hang out for a little bit after they've given up their polypeptide chain to the next tRNA. So they're empty tRNAs, and they can be excused from that E site. So 3 sites for tRNAs on the ribosome. We begin with a 30S subunit, a smaller subunit. It binds to 2 initiation factors, IF1 and IF3. IF1 is more associated with the A site. IF3 is a little bit more over towards the exit site. Once those initiation factors are in place, the small subunit can bind onto mRNA and looks for the initiation codon. Right? No surprise, that initiation codon is the start codon AUG, and we'll talk a little bit in just a minute about how it knows which of the AUG codons is a proper start codon as opposed to an other AUG codon. Okay, so we've got our mRNA in place. The AUG codon is right over the peptidyl site, and now we can bring in an additional initiation factor 2, which has GTP associated with it. So this is going to be an energy-dependent process. It's driven by hydrolysis of GTP. That initiation factor shows up in complex with that charged start tRNA. So that says F-met on it, because this is a start codon, and it's defined by this fact that it has N-formal methionine on the CCA arm, and it's got the proper anticodon CAU to recognize the AUG start codon. Excellent. That's in place. Codon-anticodon interaction, and now the initiation factor 2 hydrolyzes its GTP. That provides energy to disassociate the initiation factors, allowing the larger 50S subunit to form the complex, and now we have a complete intact ribosome able to do its work. We've assembled it, we've completed the last E site for tRNAs, and we've got our first amino acid sitting in the peptidyl center, and this one start tRNA with that N-formal methionine sitting there as well. And now we're ready to get going. Okay. So, yeah, we've got this mRNA. It's got an AUG codon right where it should be, but proteins occasionally need to have a methionine residue somewhere else in the sequence. What do we use as a codon for methionine? AUG. So here's a little bit of an ambiguity. How does the ribosome know that that AUG codon here is a proper start site? Why doesn't it just sit down at an internal AUG that's just coding for a regular methionine? The answers are pretty clear in bacterial prokaryotic systems. That AUG that defines a start codon is sitting just downstream of what's called a Shine-Delgarno sequence. So this is what these Shine-Delgarno sequences look like. Very often they have this GGAG right in there, just ahead of the start codon. GGAG, there's some variations. Here's an AAGGG, but there's pretty good correspondence to that particular sequence with GGAG at the core. What is that doing? Why does that help define the start site? Remember that the ribosome is composed both of protein and of RNA. So there can actually be Watson-Crick base pairing between the mRNA and the components of the ribosome itself. And that's exactly what's happening here. Here's our 16S ribosomal RNA. This is an intrinsic component of the ribosome. It's got a particular sequence. And look, right near where the start codon needs to park, it's got a sequence that provides good Watson-Crick complementarity for these Shine-Delgarno sequences. GGAG, okay, well, this 16S ribosomal RNA is reading out CUCC. And so that's where the mRNA is going to want to sit. And then the ribosome's job is pretty easy. Once this Watson-Crick base pairing is lined up, it just looks for the neighboring AUG. That's got to be the start codon. Great. Eukaryotic ribosomes, just like when we've been talking about other polymerases, it's quite a bit more complicated. You don't have to spend too much time and effort thinking about this. There isn't any Scheindel-Garneau sequence, at least not too often. We don't use N-formal methionine as the initiator tRNA. Instead, it's just a regular methionine. This is the composition of the mRNA as it is brought onto the small subunit of the ribosome. So it's actually not just the mRNA itself, it's already being associated with different protein factors, initiation factors, and other proteins that are going to bind to and recognize both the 5' cap and the 3' polyadenylate. And you should remember these as being really critical components of the processing of mRNAs in eukaryotes. Right? It generates a primary transcript, it modifies that 5' end, and it adds this really long string of adenylates onto the 3' end. And this is a really important reason why. These proteins recognize those, associate with them, bend the mRNA into this kind of big cyclic structure, and this is what's brought into the ribosome. These proteins are sitting in a very particular location, just upstream of the AUG that defines the start codon, and this is what tells the ribosome start at this position versus another. There can be some ambiguity here. Sometimes different start codons can be recognized, and so that will generate slightly different polypeptide products. You'll note that this initiation factor 2, just like in this case here, IF2, binding to the incoming initiator tRNA and has GTP associated with it, basically the same thing here. There is an initiator tRNA, it has methionine on it, initiation factor 2 binds, initiation factor 2 has GTP. This is also an energy-driven process, and so once we've gotten the mRNA associated with a small subunit, initiation factor 2 can hydrolyze its GTP to GDP, providing the energy to drive off the other initiation factors, and it brings in the larger ribosomal subunit to complex with the small subunit, creating the intact ribosome that's now capable of carrying out its synthesis. Okay. So a little bit more complicated in the prokaryotic system, but the rudiments are still the same. Yeah?
What is the energy from the hydrolysis of GTP used for?
It's used to kick off the initiation factors. Yeah. So there is a favorable binding interaction of the initiation factors with the mRNA and with the small subunit, and this just kind of shakes them off. Yeah.
In the case of prokaryotic ribosomes and translating proteins, does that mean that every mRNA has this Scheindel-Garneau sequence, no matter, like they all have it?
Right. So the question is, does every mRNA have a Scheindel-Garneau sequence? Essentially, they do. Almost all of them have some sequence that has favorable contacts with the 16S RNA just upstream of the peptidyl center. There can be real variations in what these look like, and some of them just barely begin to resemble the canonical Scheindel-Garneau sequence. Yep.
Initiation factors 1 and 3 are kicked off, but does initiation factor 2 stay on?
Initiation factor 2 also leaves when GTP is hydrolyzed to GDP. So the initiation factors are only around just when the small subunit and large subunit are forming the intact ribosome. We're going to need additional factors, but they're going to be elongation factors, not initiation factors. All right. So let's talk about an interesting elongation factor. So now that we have our initiation complex properly formed and an intact ribosome, we need to bring in the second amino acid. First one is in place, N-formyl thymine, it's at the peptidyl site. Let's bring in our our second amino acid. And that's going to be brought in complex, not with an initiation factor, but with an elongation factor, this EFTU. EFTU is complex to GTP, and so it brings in the amino acid into the A site and parks it there, and then GTP is hydrolyzed to GDP, and then the elongation factor can exchange its nucleotide, at which point it's ready to accept another tRNA and repeat its process of bringing in additional amino acids charged onto the tRNAs onto the A site. So, first amino acid brought into the P site by the initiation factor, second amino acid brought into the A site by elongation factor TU, also a GTP-dependent process. All right? There's a mistake in this figure that comes from a previous textbook. I'm going to let you guys look at it, figure it out, unless anybody sees it, by the way. It's a little subtle. But once you see it, it'll be up. What?
Is that GTP supposed to be GDP? When it switches from when the TS binds to the TU, right before that?
Here?
Step by step.
Here? Oh, okay. This is GDP coming off, and then a new GDP is brought in. So I think this is correct. That's not the mistake. I'll let you guys ponder it. It's not a huge deal, but once I realized it, I was kind of amused. Okay. You know, these were the pictures that we had of what's going on at the ribosome when I was studying biochemistry. And I just think it's terrific that now we have these pictures. We don't just look at like kind of blobs and ovals on these like mitt-like kind of things. We actually know exactly what the tRNAs look like as they sit in each one of these three positions. So this is the X-ray crystal structure of the prokaryotic ribosome. Here's our 50S and our 30S subunit, and here are the three sites in the ribosome. A site, aminoacyl site, the P site, the peptidyl site, and the E site, the exit site. This green little bit right here, that's our template. That's our mRNA, and you can see that the anticodons are engaging with the mRNA down here, and this must be where the business end of the ribosome is. These are the ends, the CCA arms of the tRNAs, and so this is where the bonds must be being formed. So an incoming tRNA with an individual amino acid parks at the A site, encounters the growing chain on the P site tRNA. That amino acid is added onto that chain, at which point this complex shifts down and the empty tRNA ends up in the E site, which can later be removed. We'll take a look at that. Again, here is the A, P, and E sites. Here is a blow up of just the tRNAs sitting in that ribosome. Here's the mRNA, in this case shown in yellow. And this is where we're going to make our peptide bonds. And here is our empty, used up tRNA at the exit site, leaning off towards the side, ready to be ejected. All right, let's make a bond. We're going to make an amide bond. And so this kind of arrow pushing is near and dear to my heart. I want you to know how these bonds are being made. It's easy just to draw, oh, these two beads are now together. Well, yeah, they're together, but what bond is it? How is it made? Here's our nucleophile. It's the amino group of the amino acid conjugated to the tRNA that's at the A site. It is going to make a nucleophilic attack on this carbonyl carbon that is at the very terminus of the growing peptide chain that is also appended onto the 3' end of the tRNA at the P site. When that nucleophilic attack happens, we make our amide bond and we grow that polypeptide chain by 1 amino acid. And this shows that product. So what is the leaving group of that nucleophilic displacement? Just simple organic chemistry. Nucleophilic attack, we make a bond, there's a leaving group. What's the leaving group?
The OH on the...
Right here?
Yeah.
Yeah, that OH is part of the molecule that we consider the leaving group. Yeah. Yeah, it is the entire tRNA. That's the leaving group. Okay. So nucleophilic attack, we'll take another look at this in just a second. I've got another structure. But that whole tRNA that's been sitting at the P site is the leaving group. After we've made our peptide bond, we kind of have like a sort of a hybrid structure where the tRNA itself is at the A site, the polypeptide chain is sort of leaning into the P site, and so we need to kind of shift this over before we begin the next step. But this is where we end up after peptide bond formation. This reaction, this nucleophilic attack, no surprise, it's being catalyzed. The ribosome is a catalytic entity. The catalysis is performed by the portion of the ribosome called the peptidyl transferase center. What's there? What is at the peptidyl transferase center? And I was jumping up and down about this. On Tuesday, I was so excited about it that I couldn't help but give you a preview about it. Yeah. A ribozyme? Yeah. It's a ribozyme. In other words, what is at the catalytic center? RNA. So here's a really nice view of the ribosome in which the protein is shown in blue and RNA is shown in green and in gray. We can locate the catalytic center by co-crystallizing this in the presence of puromycin. We'll talk a little bit more about that towards the end of lecture today. This is an antibiotic. It blocks up that active site, the peptidyl transferase center. So there it is. Do you see any protein anywhere around there? Hell no. It's just RNA. Amazing. Everything that's being done to perform this catalysis, this really complex catalysis with these macromolecular substrates forming these polypeptide chains, that's being catalyzed by RNA itself. And just because this is crucial, here's another picture of what's going on at that catalytic site. Here is our incoming nucleophile. Here is our growing polypeptide chain conjugated onto the terminus of the tRNA. We form this tetrahedral intermediate. How many times have we seen this? We collapse down from the tetrahedral intermediate, and here's our leaving group, the tRNA itself. and now we form this new amide bond, and we've grown that polypeptide chain by one amino acid, and the entire time it remains conjugated onto a tRNA species. We never let go of the polypeptide chain. It's always just hanging out on the tRNA. Yeah. Oh, yeah. Yeah. We're still kind of working out exactly the mechanism of catalysis. I mean, I got to be honest with you, like even for simple enzyme, protein enzyme systems, it can be a little bit ambiguous as far as like what's really going on to drive catalysis. We can create models, but we can never really be definitive about what is providing the catalytic mechanism. We come up with hypotheses. You can imagine it's really difficult to be certain about what's driving catalysis in these systems, but I'll have some figures to show you the kinds of things we think are important.
It's crazy, isn't it?
So here is the active site of the ribosome. Here we know which direction the attack, the nucleophilic attack, is coming from. We know like what are some of the important nucleobases. Here's an adenine nucleobase that seems to be engaging with the polypeptide chain that's being generated. So, you know, this is the A site right here. We know what portion of the ribosomal RNA is hanging out at that particular region. Oh, darn it. So we have like a really general notion of what's going on here. But again, what exactly is driving catalysis is not completely clear, but we do know that there's no protein around in this active site at all. So here's a really fascinating quote from one of the scientists who really helped understand ribosome structure. He said, "What is remarkable, however, is that in the ribosome, these functions are carried out largely by RNA, not protein, and this is undoubtedly the palimpsest of the origins of these mechanisms in early molecular evolution." So what is a palimpsest? Does anybody know? Yeah. The markings left behind after you erase something. Yeah. You have another idea about what that is?
Just, I would think it's an insight into how these mechanisms evolved early on.
Okay, it's actually both. So traditionally, a palimpsest is used to refer to something that's been reused. So it's been erased and reused to provide new meaning. And in this case, this reuse gives us some idea about what we think were the original pieces of machinery in very early life forms, the very first steps of molecular evolution. So let me explain this a little bit more. What is a palimpsest? Back in the good old days, when somebody wanted to create a text, whether it was copying down a piece of philosophy from Archimedes or a piece of Holy Scripture, they needed something to write on. And so they could use like sheepskin or papyrus, but this stuff was precious. You can't just go over to Staples and buy a new ream. So when you had something like a piece of sheepskin that you were drawing on, and you thought, no, I want to write something new, sometimes you wouldn't have a nice blank piece of sheepskin lying around, and so you would go to your previous text and you would just scrape it off, try to remove all the ink, and reuse it, and write something else on it. So lots of times in archaeology or studying ancient texts, you can see not only what was most recently written, but in some cases you can see the traces of the original document, the original text that was put on to that parchment or papyrus. So that's what some of these things look like. And now this term, palimpsest, is used more or less in the meaning that you implied that if you look at some archaic structure, you can see the original use for that structure and then see how it was embellished or repurposed or partially destroyed and rebuilt to provide for a new function. So what does this mean then in this sense of the ribosome being a palimpsest? It means that this is giving us an indication of some of the important catalytic activities back in the very first steps of life, that this must be one of the crucial aspects of biological polymer synthesis, and it's being carried out by a ribosome in which the RNA is the catalytic entity. And a lot of people, such as Harry Noller, think that this is telling us that RNA was the initial catalytic entity before proteins in the form of enzymes took over that role. Interesting concept. Okay, so let's continue making our polypeptide chain. The next step is going to be using a different elongation factor. This is elongation factor G. This comes in and shifts everything over to locate the tRNAs into their new sites on the ribosome. So, okay, we have got this tRNA as our leaving group. It no longer needs to choke up the P site. We can begin to chuck it out. So we're going to shift it over to the E site, and now we're going to relocate the tRNA that was at the A site and locate that tRNA with its growing polypeptide chain at the P site. That frees up the A site, and now a new charged tRNA can come in. That shifting, that translocation, that movement of the ribosome down the mRNA template and the shifting over of the tRNAs into the other sites on the ribosome, that is a GTP-dependent process that is carried out by elongation factor G. And you see that ribosome moving down the mRNA template. Here's another picture of it, just showing where the tRNAs are associating with the mRNA, where the amino acids on the tRNAs are interacting with the peptidyl center. This shows them kind of leaning over as the peptide bond is being generated. And then here's our elongation factor G that now swings the tRNAs into their new positions. tRNA is dissociated from the E site, and we have an empty A site. And we just repeat this process throughout the course of elongation until we've completed our polypeptide chain. How does the ribosome get it right? What do we mean by getting it right? So elongation factor TU, I've told you that it's a GTP-dependent process, right? I'm talking about that. Yes, elongation factor TU, this GTP-dependent process, as it brings in the new charged tRNAs, it can do a little bit of checking. It can make sure that it's getting it right. And what does that mean? It's actually looking for proper codon-anticodon interactions. The identity of the amino acid on the tRNA is not checked. So it's just looking to see whether or not the codon on the mRNA is matched up with the anticodon on the tRNA. As long as that's in place, the EFTU will hydrolyze the GTP, exit, and leave that tRNA in place at the A site to function as the nucleophile to make the next amide bond. And so this is what the EFTU is looking for. Proper distance between the nucleobases on the codon and anticodon, as long as it's got that right pairing up. This portion of the 16S RNA on the ribosome will fall into place. The EFTU will see that it's snugly fit in here, and the EFTU, as I said, will hydrolyze its GTP, do its job, and leave the tRNA there. Okay, so we make our polypeptide chain, we repeat those steps over and over, continue making the amide bonds. Now we get to the end of our message. And how do we know it's the end of the message? Yeah. There's a stop codon. Right? So there's, we said 64 different triplet codons, 3 of them encode a stop. Here's one of them, UAG. And there's no tRNA that corresponds to that codon. Instead, a protein release factor likes to bind onto that UAG site when that UAG is at the A site in the ribosome. This allows for a new kind of reaction to happen at the peptidyl center. It's a hydrolysis reaction, and it breaks the ester linkage between the polypeptide and that 3' end of the tRNA that's sitting at the P site. It's at this point that the polypeptide is released, and only at that point. The hydrolysis reaction liberates the polypeptide chain. And now the large subunit can disassociate from the small subunit. All the different components can come apart. The mRNA can be released to be reused. And the 2 subunits can also reform in response to a new mRNA with its start codon. Okay? All right, so this is a chemical reaction, right? What is the nucleophile in this case? Previously, as we were making amide bonds, the nucleophile was the amine of the amino acid conjugated onto the tRNA. Yeah.
Is it the carboxyl?
Nope. It's a hydrolysis, so the nucleophile presumably is water. Right. So water is generally excluded from the peptidyl center. And one big thing that this release factor can do is allow water to come into that peptidyl center. That acts as the nucleophile, and because that's a free molecule, once it acts as the nucleophile, boom, ester bond is cleaved, the polypeptide chain is released. And this just shows another representation of the release factor at the A site that permits water to come in and perform that hydrolysis reaction, liberating the polypeptide. And again, like this is the way we used to think about it. And now that we have actual structures, we can see the release factor 2 bound into the A site, recognizing the termination codon. Pretty cool. Great. So we've completed our polypeptide synthesis. Yeah. The ribosome release factors recognize the stop codons and then disassociate the protein chains. And that happens because the stop codons have no tRNA in normal cells. That kind of brings up an interesting question. What if you artificially introduced a tRNA that was complementary to a stop codon? That would be kind of amazing. It's been done. And if you charge an amino acid onto the CCA arm, create a completely new anticodon that recognizes a stop codon, you can overwrite a stop codon and get some completely bizarro amino acid to sit there. We've rewritten the genetic code. But this just proves the point that there are no tRNAs that are responsible for recognizing those stop codons. Release factors do that, but we can provide an exogenous one. So let's take a complete look at this process. I think this is one of these cases where an animation is going to be really helpful. This is a nice one that was made shortly after the structures for the ribosome were elucidated, and I'm very sorry that we have no music to go along with this. Okay, so here's our mRNA. It's associated with a small subunit, 30S subunit. Here is our N-formal methionine tRNA. Now that that's in place, the large subunit comes in, and the additional initiation factors are released. Now we're moving on to elongation. We need to bring in a new tRNA in complex with elongation factor TU. There's our GTP hydrolysis. Okay, it must have figured out it was proper codon-anticodon. It likes it and lets that tRNA walk its amino acid over to the peptidyl site. Now that bond has been made. Once it does that, it shifts that tRNA leaving group over towards the E site. Elongation factor G comes in, hydrolyzes GTP, pushes everything over, and now elongation factor G will leave, and we're ready to start the next cycle of elongation. tRNA leaves from the E site, and now let's see it all going down. One after the other. mRNA being fed in through the cleft between the large subunit and small subunit. And what's coming out the other end? It's the growing polypeptide chain. And it's coming out of a tunnel in the large ribosomal subunit. Here comes our polypeptide out of the tunnel. And here comes the release factor. And now everything comes apart to be reused. All right. Wow.
There's no excuse for what they did to you. It's a hate crime.
Based on a true story
I created a world where I can heal.
of a man who turned tragedy
being prejudiced against for my ethnicity.
That's very ignorant for you to say.
I'm tempted to show you guys that again, but instead I'm going to post this link on the course website, and I recommend that you check it out there. Okay. What's up in that tunnel? That's kind of interesting.
Wow, that's crazy.
This shows the tunnel in the large subunit. Again, we have an atomic resolution picture of that. So we know the size of that tunnel, and we can model different kinds of protein structures in there. If we model in an alpha helix, it fits really nicely in that tunnel. But if we try to fit in like a complete domain of a folded protein, it won't fit. The conclusion then is, at what point are proteins folding as they're being synthesized in the cell? Possibly they're beginning to fold even as they're being nascently generated on the ribosome itself, but only to a limited degree. We might be able to generate little bits of secondary structure. We're not going to be able to fold entire domains, at least not until the polypeptide chain is completely ejected from the ribosome. Another little cartoon of how peptide bonds are being formed, and this just shows a nice set of codons so that you can see the incoming tRNAs forming the correct codon-anticodon association, and then walking itself down towards the stop codon where there's cleavage and release. I love this cartoon. I recommend you spend a lot of time with it because it really breaks the whole process down into multiple steps. The fact that there needs to be codon recognition by an incoming tRNA in complex with EFTU. That where there's proper codon-anticodon interactions, there can be activation of the hydrolysis of GTP within that elongation factor. That drives the hydrolysis of GTP. If that is conducted properly, there's something called an accommodation event that swings the amino acid over onto the peptidyl site. If there is improper codon-anticodon interaction, that tRNA is rejected and we start over here. But then we make our peptide bond, elongation factor G comes in, also GTP hydrolysis event, allowing the ribosome to be translocated, elongation factor G is released, and then we repeat the whole process. So it looks like 2 GTPs are hydrolyzed for each round of this event. This is a cool image of protein chains being synthesized on an mRNA in a bacterial cell. And you can see that just one mRNA is being processed by multiple ribosomes. So this is termed a polysome, and this just indicates that the same mRNA can be used over and over again to crank out multiple polypeptide sequences. This is simultaneous transcription and translation of a DNA sequence. Multiple ribosomes sitting on that mRNA as it's being generated from the DNA template. This we only see in prokaryotic systems. Why don't we see it in eukaryotic systems? Yeah.
Eukaryotic mRNA has introns, need to get spliced out.
Right. So the eukaryotic mRNA needs to be processed, and it's physically made in a different location from where the ribosomes are hanging out. All right, I'll just end up today by telling you that the textbook contains some interesting information about antibiotics. It's of passing interest that many antibiotics target the ribosome itself. In particular, this puromycin does a really cool thing by sitting in the A site and capturing the polypeptide chain and preventing it from being extended. You can take a look at that. And then we need to complete this picture by just realizing that once the polypeptide chain is being made by the ribosome, it needs to fold up. In some cases, it can do that on its own. In other cases, it needs additional machinery. We talked about that before. Next time, we will talk about post-translational modifications, how that is tied into the programmed degradation of proteins. And we will have our next episode of Meet the Chemist, because this guy, Professor Neil Shah, is coming down from Columbia University to talk to you guys about a special case of post-translational modification with phosphorylation of tyrosines, and he's an expert on that topic, so he'll come and talk to us about this. Here's our recap. Understand what controls fidelity and translation. Remind yourself the 2 chemical reactions that occur on aminoacyl tRNA synthetases. Know all of those steps at the different tRNA sites at the ribosome. Know those bond-forming events. Be able to draw those nucleophilic attacks. Realize that the ribosome is a ribozyme. And then think about initiation, elongation, and termination as it happens on the ribosome. All right.
Yeah, bro, you think?